for decades, biologists looked at the fat cells in your body and basically thought they thought they were biological addicts.
Yeah, just kind of these dumb, inert storage tanks.
Right, like where your body just shoves away excess calories to deal with later. But, if you actually zoom into the cellular level, those spherical fat blobs, they're called lipid droplets, they are not quiet at all.
Not even a little bit.
They are these bustling, highly regulated metabolic factories. And if the intricate GPS system that directs molecular traffic to those factories breaks down, the consequences for you aren't just like carrying a bit of extra weight.
No, it's way more serious. You end up with severe, life-threatening metabolic disorders.
Exactly.
We are talking about conditions like MASLD, right? Which stands for metabolic dysfunction-associated steatotic liver disease.
Which is a massive global health issue right now.
Oh, absolutely massive. The health of your liver and really your whole metabolic system completely depends on these highly specialized worker proteins navigating through the cell to find and operate on those lipid droplets.
So today, we're taking a deep dive into a really fascinating paper from Nature Cell Biology. It actually answers a massive biological mystery about this exact process. Like, how do these specific metabolic enzymes navigate the microscopic cellular highways, recognize these fat droplets, and, actually stick to them?
Yeah, and to really grasp the scale of this commute, you have to imagine you are one of these worker proteins.
Okay, let's unpack this. I'm a protein.
But you're a protein. You are synthesized in this massive labyrinth-like factory inside the cell called the endoplasmic reticulum, or the ER.
Okay, the ER.
Which brings us to the very first architectural hurdle. The ER is surrounded by a traditional bilayer membrane.
2 layers.
Exactly. If you picture a standard cellular wall, you are picturing a bilayer. It's 2 layers of fat molecules called phospholipids stacked tail to tail. It's super stable.
It's the standard building material.
Right, but the lipid droplet, the LD, is an architectural anomaly. It does not use standard building materials.
Which is so wild. Let's break down why that is, because the physics of it are just fascinating.
The physics are totally different.
Yeah. So the lipid droplet has a core made entirely of neutral lipids. This is mostly triglycerides and cholesterol esters.
Right. Pure fat.
Pure fat. These are intensely hydrophobic materials. They violently repel water. So because you have this massive core of pure oil sitting in the watery soup of the cell, you can't wrap it in a standard 2-layer membrane.
No, because the inner layer of a normal membrane would chemically clash with that oil core. It would fall apart.
So the cell wraps the lipid droplet in a monolayer, just a single layer of phospholipids.
And that structural difference between those two environments, it cannot be overstated. When a vital metabolic protein is born on the ER, it's embedded in this nice, stable 2-layer sheet. But to do its job, it has to physically travel to the lipid droplet and embed itself in a single, slippery layer of fat.
If we think about the ER as a massive, familiar 2-lane asphalt highway...
Oh, I love this analogy.
Thanks. So transitioning your protein vehicle onto the lipid droplet is like driving off that solid highway directly onto a bizarre 1-lane monorail suspended over a literal lake of oil.
That's exactly what it's like. It's an utterly alien environment.
Right. And it poses a major question for you, the listener. If you had to design a vehicle to drive seamlessly from normal asphalt straight onto an oily monorail, what kind of specialized wheels would you even need?
And how do proteins even make that transition? And once they get there, why do they stay put instead of just washing back and forth?
So to figure this out, scientists had to actually watch the proteins make the commute.
Which is way harder than it sounds. Historically, tracking a single molecule inside the chaotic, incredibly dense soup of a living cell has been basically impossible.
You're usually just looking at, like, blurry blobs glowing on a screen.
Exactly. But in this paper, the researchers used this insane tracking technology called MIMFLUX nanoscopy, and they paired it with a machine learning tool called DeepSPT.
DeepSPT, okay.
Yeah. They tracked a few crucial metabolic proteins like GPAT and HSD17B13, and a model cargo protein they called live drop.
Live drop, I like that.
Right, and they tracked them with sub-15 nanometer resolution.
Wait, 15 nanometers? That's staggering.
It's unbelievable. For perspective, a single strand of your DNA is about 2.5 nanometers wide.
Wow.
So they were watching single proteins navigate this landscape, and they were sampling at 3.5 kilohertz.
Which means, what, they were taking thousands of snapshots a second?
3,500 snapshots a second.
That's crazy.
They gathered over 2 million individual data points just mapping the precise paths of these proteins.
So they basically had a microscopic high-speed camera.
Exactly, catching a bullet mid-flight. And because they had that level of temporal resolution, they could measure the exact speed of the proteins as they moved along the ER highway and compare it to their speed on the lipid droplet monorail.
Okay, but my immediate assumption here is that if a protein is cruising along a standard two-layer membrane and then suddenly hits a super dense, weird monolayer packed with triglycerides, it has to hit a wall.
Right, you think so.
Like the physics dictate it should slow down drastically, like a runner suddenly hitting waist-deep mud.
That is exactly what everyone expected. But what's fascinating here is the data showed the exact opposite.
Wait, really?
Yeah. The overall speed of the proteins, they measure this as the apparent diffusion constant, or DAP. It was completely identical in both the ER and the lipid droplet.
Identical.
Basically identical, around 0.078 to 0.085 square micrometers per second in both places. They didn't slow down by a fraction of a second.
So they hit the weird oil monorail and just maintain their sheer velocity?
They maintain the exact same speed.
But if they aren't slowing down at all, how do they accumulate? Shouldn't they just fly right off the other side of the droplet?
Well, they don't fly off because of a physical phenomenon known as anomalous diffusion.
Anomalous diffusion. Okay, explain that.
So while the pure speed remains unchanged, the style of the movement fundamentally shifts.
Speed versus style.
Yes.
Yeah.
There's a metric called the anomalous diffusion exponent alpha. On the ER, this alpha is around 0.61.
Which means what, practically?
It means it's closer to free, Brownian motion. The proteins are roaming, making long, sweeping runs, exploring wide stretches of the highway.
Okay, got it.
But the moment they hit the lipid droplet, their behavior changes mathematically. The alpha drops down to 0.38. The area they are able to explore shrinks dramatically.
Okay, so think about this for a second.
Yeah.
You're sprinting down a wide-open football field, making these long, sweeping runs. That's the protein on the ER.
Right.
Now you transfer to the lipid droplet. You maintain that exact same intense sprinting speed, but suddenly, instead of running in a straight line, you are trapped running furiously in a tiny, tightly confined circle pit.
That is the perfect way to visualize it. They become nano-confined. They are moving rapidly, but they are trapped within these incredibly small boundaries on the surface.
Okay, let's do a quick map check for everyone listening, just to solidify where we are in this journey.
Good idea.
So, bullet 1. Lipid droplets are these unique single-layer fat storage hubs, and getting proteins delivered to them properly is vital for preventing diseases like MANS-LD.
Bullet 2. Advanced tracking like MINE-MFL-UC shows us that proteins surprisingly move at the exact same speed on the ER and the lipid droplet.
And bullet 3. Even though they don't slow down, proteins on the LD experience anomalous diffusion, meaning they get trapped in tiny areas, shifting their movement from free roaming to tightly confined circle pits.
Exactly.
But if they are running in a circle pit, something physical has to be keeping them there. They can't just be running in circles for no reason.
No, they can't.
There has to be an invisible fence.
And that invisible fence is a structure we call a nanodomain.
Nanodomains.
Right. When we picture a lipid droplet, we usually think of a perfectly smooth, uniform sphere of fat. But it isn't uniform at all.
It's textured.
Yes. The surface is speckled with tiny, highly dense patches. These are the nanodomains. They're incredibly small, averaging just 0.0032 square micrometers.
That is so small, it's hard to even conceptualize.
I know. But when the high-speed tracking data plotted the path of proteins, they saw that the moment a protein entered one of these specific nanodomain zones, its mobility, like its ability to roam, dropped by 50%.
Oh, wow.
They clustered tightly together in these microscopic patches.
But the protein doesn't have brain. It doesn't have eyes to see the nanodomain, so there has to be a chemical interaction pulling it in, right? It needs a biological password.
Exactly. And the researchers hunted for that password. They looked at the live drop sequence, which is a hairpin-shaped part of the GPAT4 protein.
The live drop hairpin.
Right. They started looking at the specific sequence of amino acids that make up that hairpin, trying to figure out which ones were acting as the key.
I'm guessing that if you locate the specific amino acids that act as the key and you chemically mutate them, the protein would hit the droplet and just slide right off.
That is exactly the experiment they ran.
Really?
Yes. They created mutant versions of the protein. So the normal wild-type live drop would arrive at the lipid droplet, interact selectively with the nanodomains, and cluster tightly.
Working perfect.
Working perfectly. But then they made the 3W mutant. They specifically mutated the bulky, water-repelling amino acids known as tryptophans. They changed 3 of them.
So they messed with the password. They removed the sticky parts.
They altered them, and the result was striking. The newly mutated protein still physically arrived at the lipid droplet.
But it couldn't stick.
It completely lost its grip. The area it explored increased by 80%. It was just wandering all over the surface, unable to cluster properly.
It couldn't get trapped in the circle pit.
No. And when they took it a step further, creating the 3W plus KRR mutant, where they scrambled the sequence even more to remove the positively charged residues, the protein couldn't even target the lipid droplet in the first place.
It just floated away.
It never even arrived.
Here's where it gets really interesting to me, looking at the underlying lipid chemistry. Why is tryptophan specifically the magic password?
Well, it's because tryptophan specifically interacts with the ester groups of the triglycerides.
The fats making up the massive core of the droplet.
Right, and because it's a single-layer membrane, those triglycerides are highly exposed.
You can almost think of the tryptophan amino acids as pieces of high-affinity molecular Velcro.
Oh, that's good.
Right. So the 2-lane highway of the ER membrane doesn't have the right fabric for the Velcro to stick to. The protein just glides over it.
It ignored it.
But the lipid droplet monolayer is coated in the perfect fuzzy material, those exposed triglycerides. So the protein flies onto the droplet, the tryptophan Velcro catches the triglyceride fuzz, and bam, it instantly gets retained in these tiny nanodomain circle pits.
Yes, and it reveals this beautiful, elegant system where the cell uses the local chemical environment, the physical properties of the fats themselves, to physically dictate how a protein behaves and where it is allowed to accumulate.
Okay, let's do our second quick recap.
Bullet 1. Proteins get confined on the LDs by locking into tiny patches called nanodomains.
Bullet 2. This locking mechanism relies on specific amino acids, like our molecular Velcro, tryptophan, that bind to the LD's unique, fuzzy lipid environment.
But if we pull back and look at the whole system, there is still a massive physical link missing here.
Right. We know how they move on the ER. We know how they Velcro themselves to the droplet once they arrive.
But how do they cross the literal gap between the ER bilayer and the LD monolayer in the first place?
Yeah, they aren't just jumping across empty cellular space. And if the ER is directly touching the lipid droplet, I mean, how do you go from a double layer to a single layer without the whole system rupturing and leaking pure oil everywhere?
You'd think it would pop like a balloon.
Exactly. There has to be a highly controlled junction.
There is.
Yeah.
And it relies on stable membrane bridges.
Bridges.
Yeah, the cell uses a specific structural protein called seipin, S-E-I-P-I-N. Seipin proteins group together to form this highly stable ring complex right at the exact physical contact points between the ER and the lipid droplet.
Like a toll booth.
Exactly like a toll booth or a tunnel.
The structural engineering of that is brilliant.
And the researchers actually proved this using that DeepSPT machine learning tool we mentioned. They plotted the exact spatial coordinates in the cell where the live draw proteins switched their behavior. They looked for the precise microscopic location where the protein stopped making long sprinting runs and started doing the tight circle pit sprints.
The moment they crossed over.
Right. And those transition points perfectly, and I mean perfectly, colocalized with dense clusters of seipin.
Wow. So seipin is the actual gateway.
It is the physical bridge.
So what happens if you blow up the bridge? Like if you create a seipin knockout cell that entirely lacks that structural protein, does the traffic just pile up on the highway?
In the knockout cells, the results were catastrophic for the traffic.
Yeah.
Many of the lipid droplets completely detached from the ER.
They just drifted off.
They floated off as independent islands. And they received absolutely zero of the live drop worker proteins.
That makes total sense. Seipin isn't just a random hole in the wall. It's the Golden Gate Bridge connecting two entirely different landmasses.
Exactly.
If you blow up the bridge knockout seipin, the cargo simply cannot cross over to the island no matter how much Velcro it has.
Without seipin maintaining that contiguous connection, the vital metabolic trafficking entirely halts.
Okay, but this brings us to a huge structural question. We know seipin acts as the Golden Gate Bridge, but what kind of bridge is it?
What do you mean?
Well, once a protein crosses that bridge and its Velcro catches on to the lipid droplet, is it banished there forever? Is this seipin bridge actually a one-way trap door where proteins go to do their job and they can never leave?
Oh, this is my favorite part of the paper. To figure out if it was a one-way street or a two-way street, they designed an incredibly clever test called a reverse RUSH assay.
Reverse RUSH.
Yes. It's basically a highly controlled catch-and-release experiment, done in 3 distinct steps.
Walk us through it.
So step 1, they use a chemical called biotin to artificially release a massive wave of proteins all at once from the ER.
Like opening a floodgate on the highway.
Yes, allowing all the proteins to rush over the seipin bridge and just deeply accumulate on the lipid droplets.
So you force the lipid droplet to become fully loaded with workers.
Exactly. Then step 2 is locking the system down. They flood the cell with avidin.
Avidin. Which does what?
Its sole job is to hunt down and sequester all the biotin. It neutralizes the initial release chemical. So now the proteins are just sitting on the lipid droplet, operating under normal biological conditions.
Okay, so the floodgate is closed. What's step 3?
Step 3 is the trap. They engineer a biological hook and leave it waiting back on the endoplasmic reticulum.
Oh, wow.
Right. If any protein manages to leave the lipid droplet and cross back over the bridge, the hook will catch it and hold it in place.
You set a snare back at the start line. So if it's a one-way trap door, the hook catches nothing, right? Because the proteins are stuck on the lipid droplet.
Right.
So what did the hook catch?
When they checked the cells after leaving them overnight, almost all of the live drop proteins had returned to the ER.
Wait, really? Almost all of them?
Almost all of them. They were caught by the ER hook. And the single-molecule high-speed cameras literally caught individual proteins moving backward across those same seipin bridges.
That is mind-blowing.
They watched them transition from that tightly confined Velcro-locked movement on the LD back to free, sweeping movement on the ER highway.
Okay, let's pause and really consider what this means for how our bodies function. If we connect this to the bigger picture, it completely shatters the idea that organelles are permanent, dead-end destinations.
It changes everything. It proves that accumulation on the lipid droplet is not driven by a permanent physical barrier or a closed gate keeping them inside.
It's driven by local retention.
Yes. The proteins are absolutely physically capable of leaving whenever they bump into a seipin bridge. They just usually don't, because the chemical Velcro of those nanodomains holds on to them with such high affinity.
It's an affinity system, not a prison system.
Exactly.
The connection is a continuous two-way street. Proteins accumulate on the fat droplet simply because they chemically prefer to be there. And if you think about why a living organism would evolve this way, why a cell might prefer a two-way street over a one-way trap door, it comes down to metabolic flexibility.
Oh, 100%.
Think about your own body. You don't have the same metabolic needs when you are sleeping as you do when you are running a marathon or, fasting.
Right. Your needs change constantly.
So if your cell suddenly needs to recall all those metabolic enzymes from the fat droplet to stop processing fat and do something else, a two-way bridge allows the cell to reorganize its molecular workforce instantly.
It doesn't have to build new proteins from scratch. It just lets them flow back over the bridge.
It's so efficient.
And this is where we really start to see the universal rules of biology emerge. The research has highlighted that this mechanism, this lipid protein nanoclustering, it isn't just a weird trick used by fat droplets.
Really, it happens elsewhere.
Yeah. It closely mirrors how protein sorting works in other parts of the cell, like the secretory pathway.
Oh, like using sphingolipids and cholesterol rafts.
Exactly. It reveals a fundamental design principle. The cell doesn't always build solid walls or trapdoors to organize its workers. It uses the diverse physical and chemical properties of fats themselves to sort, organize, and direct traffic.
We've learned today that biology uses the actual physical shape and chemistry of lipids to build temporary cages, these nanodomains that hold vital enzymes exactly where they are needed, using a nanoscale Velcro system.
It's a beautiful piece of biological engineering.
It really is. But I want to leave you, the listener, with a final thought to mull over. We've talked about how this Velcro requires a very specific lipid environment to stick. But the lipid composition of your cells is not static.
No, it definitely isn't.
The types of fats circulating in your body change drastically based on what you eat, your stress levels, your overall environment. So what happens to this delicate, highly tuned chemical Velcro system when your overall metabolic state shifts?
That is the million-dollar question.
If your cellular fabric changes because of a prolonged poor diet, if the ratio of triglycerides in your droplets physically shifts, are your worker proteins still finding their way home? Or is that subtle change in the lipid fuzz the exact moment the microscopic traffic in your liver starts to break down?
And that right there is the exact frontier of metabolic research. Understanding this nanoscale traffic finally gives us the tools to investigate it.
Keep diving deep, keep questioning the invisible microscopic worlds operating inside you right now, and we'll catch you on the next deep dive.